And we left on this one. I know at the end I was pretty fast with my talking there, but I had to actually reset it. But basically, what this is showing is there can be this sexual cycle that's different between animals and some plant algae. And then again, to reiterate a little bit more, this is the fungi and some protists. Um, The only diploid stage is a single cell zygote, which is different. There is no multicellular diploid stage. The zygote produces haploid cells by meiosis. So in this particular organism, the single cell is diploid, but the unicellular or the multicellular is haploid, and the haploid reproduces by mitosis. The haploid adult produces gametes by mitosis, and depending on the life cycle, either a haploid or diploid can divide by mitosis. Only a diploid can undergo meiosis. And this makes sense because if you have a diploid, so a diploid which goes through a meiosis, if you have two diploid cells getting together with regard to fertilization, what you eventually are going to have is just going to have way too much genetic material. So when it makes sense, you're always in this process of fertilization, it's always going to be a process of reduction. So reducing the number of chromosomes so that those half reduction number of the haploid then can get together with other haploid and create a diploid. So the 3 cycles, the halving and doubling of chromosome contributes to this genetic variation. This is just an added slide. We have introduced a lot of terms, and some of the terms were introduced last chapter. But this idea of sister chrome, I guess we'll start at the upper left. The centromere, remember that's a little waist area between two homolog chromosomes or sister chromatids. And then you have the kinetochore. Those are the proteins that the microtubules are going to attach to. And then you have non-sister chromatids. So what they're represented by the 2 different colors here. You have a duplicated chromosome. What you're seeing is a pair of homologs that are ready to be split up in this process called meiosis. And then sister chromatids, sister chromatid cohesion. We talked about that a little bit. We're going to introduce a slightly different or an additional idea to cohesion. So those sister chromatids are pretty much attached to each other so that all the loci, all the alleles line up. And then you still have, and that's one way that they're attached, but they're also attached via these centromeres. This is a new term, this chiasma is a new term. We'll talk about it a little bit more, but it's a place where you have fragments of DNA or broken DNA that then can attach to the broken DNA on a sister chromatid. Or a non-sister chromatid, I guess you might say, of the haploid cells that are going to merge in this process that we're going to explore called meiosis. So it's just a review here, and if this works for you, then print it out and look at it with regard to where the key players are in this process of mitosis and also meiosis. Like mitosis, meiosis is preceded by replication of chromosomes. So you start out with this germ cell or this precursor cell. We call it a it depends on whether you're referring to a spermatocyte or spermatogonia is what it's called in the human male, but we'll just call it a germ cell, is going to replicate chromosomes. So that's going to be a diploid cell replicating to become another diploid cell. But what happens, one of the reasons why it happens that way in humans is that original diploid cell, which replicates itself, can then stay and replicate again. But meiosis takes place in two sets of cell division. We call it meiosis 1 and 2 because it's very, very important you keep that straight whether you are in meiosis 1 or meiosis 2. Two cell divisions result in 4 daughter cells rather than 2 daughter cells that you have in mitosis. So what happens, just kind of to fast forward, but we'll flesh this out individually, is there is a replication of the chromosomes before meiosis 1 starts, but there is no replication before meiosis 2 starts. And that way you actually have this reduction of chromosomes. Each of the daughter cells has only half the complement, half the chromosomes as each parent. Meiosis 1 reductional division, homologs pair up and separate, resulting in 2 haploid daughter cells. And the reason we call them haploid daughter cells is they do have a cohesion of 2 chromatids together, but they're identical chromatids. So that it's not a diploid cell because you have 2 of a haploid. And so we still call them haploid daughter cells with replicated chromosomes. In meiosis 2, you have another division, equational division, where the sister chromatids separate. The result is 4 haploid daughter cells with unreplicated chromosomes. So that those chromosomes that are in the 4 haploid daughter cells all came from that precursor cells. That's where the original chromosomes came from. What this does is allows for a lot of variety, and it also is all done by independent assortment. So from your mother and father, just given the chances of variety, I think your book talks about something like 8 million different variations can take place just based on this, the fact that you have 23 chromosomes that you're going to get from your mother and your father, and they can actually get together in so many different varieties of those 23. And so what you see over here, something that we're familiar with, interphase, that's where the chromosomes are going to replicate. Meiosis 1, you still have homologs, but they are going to be, the homologs chromosomes separate, and now you have duplicated chromosomes. So it's a haploid cell, duplicated chromosomes, and then meiosis 2, you have those sister chromatids separating. So all this image is showing you here is how the numbers between meiosis 1, where you have reductional division, and meiosis 2, the equational division, where the numbers actually go from diploid to a replication haploid to breaking apart that replication haploid into the sister chromatids separate. It kind of, you have to look at it a few times, but once you look at it, I think it'll get into your head. This is a nice picture that you can't see very well here on this slide. However, you have a great double page in your book, which I invite you to look at. It goes through each of the stages and tells you a little bit about what's happening in each stage. And when you look at that, so starting it with prophase 1, There is certain boldface words that are very, very useful to know. In prophase 1, you have, as we talked about, replication. And in prophase 1, you have replication of two pair of homologs. And because of this, you have the capacity for a process known as crossing over. Crossing over occurs in prophase 1. Right now, think about it in the fact that you have so much genetic material in a very, very small space that it would be kind of easy for some of that material to be transferred one chromosome to another. It's not quite that simple. It's a little more organized than that, but you can think about it that way at this time. And the regions where crossing over can take place are called, or has taken place, I guess I might say, are the chiasmata. Sorry. And then late prophase 1, microtubules are still going to attach to the kinetochore and line these, start lining these chromosomes up. In metaphase 1, the pairs line up. They separate in anaphase 1. They are still pairs of that sister pairs of sister chromatids. At that time with some variation. And then in telophase 1 and cytokinesis, the pairs separate. In prophase 2, you have a new spindle fiber made, a kinetochore attached. They are lined up down the middle. Now you have the identical haploid sister chromatids. And then you have, well, the non-identical, I guess you might say. They are then going to line up down the middle, separate in anaphase, and then go through telophase 2 and cytokinesis, and you're going to have 4 cells there with the haploid complement that is completely different than the original precursor cell. And you have part of the image here in terms of what's happening in each of them, and then I will repeat it again just because we want to go into it in a little bit more detail. Prophase 1 occurs more than, it's about 90% of the time required for meiosis. It's the big part of the event. In that, chromosomes condense. A little bit review. It's a repeating a little bit of what we just said, but it's okay. We can hear it again. There is something new that's happening here in this prophase 1. It's called synapsis. And those homologous chromosomes align gene by gene. And this is not only the cohesion of the homologs, but also the homologous chromosomes. So you're actually going to have 4 sets line up, or 2 pair. So 4, and they're also going to line up. So we have cohesion, but you also have this other system that causes this synapsis, this other adhesion. So you actually have what's known as a tetrad at that time, where you have the 2 pair that you got from your mom and your dad lining up with another 2 pair and forming a group, all of them together, the tetrad. And what happens there is, so the chromosomes form a tetrad, a group of 4 chromatids. You have that word there. Each tetrad is usually one or more, what you have is one or more regions where their crossing over can take place. We're going to have to pick this up on the next one.